Embryonic stem cell, Wikipedia audio. Embryonic stem cells are pluripotent stem cells derived from the inner cell mass of a blastocyst, an early-stage pre-implantation embryo. Human embryos reach the blastocyst stage 4-5 days post-fertilization, at which time they consist of 50-150 cells. Isolating the embryoblast or inner cell mass results in destruction of the blastocyst, which raises ethical issues, including whether or not embryos at the pre-implantation stage should be considered to have the same moral or legal status as embryos in the post-implantation stage of development. Human ES cells measure approximately 14 μm while mouse ES cells are closer to 8 μm. Embryonic stem cells, derived from the blastocyst stage early mammalian embryos, are distinguished by their ability to differentiate into any cell type and by their ability to propagate. Embryonic stem cells' properties include having a normal karyotype, maintaining high telomerase activity, and exhibiting remarkable long-term proliferative potential. Properties: embryonic stem cells of the inner cell mass are pluripotent, that is, they are able to differentiate to generate primitive ectoderm, which ultimately differentiates during gastrulation into all derivatives of the three primary germ layers, ectoderm, endoderm, and mesoderm. These include each of the more than 220 cell types in the adult body. Pluripotency distinguishes embryonic stem cells from adult stem cells found in adults. While embryonic stem cells can generate all cell types in the body, adult stem cells are multipotent and can produce only a limited number of cell types. If the pluripotent differentiation potential of embryonic stem cells could be harnessed in vitro, it might be a means of deriving cell or tissue types virtually to order. This would provide a radical new treatment approach to a wide variety of conditions where age, disease, or trauma has led to tissue damage or dysfunction. In 2012, the Nobel Prize for Medicine was attributed conjoint to John B. Gordon and Shinya Yamanaka for the discovery that mature cells can be reprogrammed to become pluripotent. Additionally, under defined conditions, embryonic stem cells are capable of propagating themselves indefinitely in an undifferentiated state and have the capacity, when provided with the appropriate signals, to differentiate, presumably via the formation of precursor cells, to almost all mature cell phenotypes. This allows embryonic stem cells to be employed as useful tools for both research and regenerative medicine, because they can produce limitless numbers of themselves for continued research or clinical use. Because of their plasticity and potentially unlimited capacity for self-renewal, embryonic stem cell therapies have been proposed for regenerative medicine and tissue replacement after injury or disease. Diseases that could potentially be treated by pluripotent stem cells include a number of blood and immune system-related genetic diseases, cancers and disorders, juvenile diabetes, Parkinson's disease, blindness and spinal cord injuries. Besides the ethical concerns of stem cell therapy, there is a technical problem of graft versus host disease associated with allogeneic stem cell transplantation. However, these problems associated with histocompatibility may be solved using autologous donor adult stem cells, therapeutic cloning, stem cell banks, or more recently by reprogramming of somatic cells with defined factors. Embryonic stem cells provide hope that it will be possible to overcome the problems of donor tissue shortage and also, by making the cells immunocompatible with the recipient. Other potential uses of embryonic stem cells include investigation of early human development, study of genetic disease, and as in vitro systems for toxicology testing. According to a 2002 article in PNAS, human embryonic stem cells have the potential to differentiate into various cell types, and, thus, may be useful as a source of cells for transplantation or tissue engineering. Current research focuses on differentiating ES into a variety of cell types for eventual use as cell replacement therapies. Some of the cell types that have or are currently being developed include cardiomyocytes, neurons, hepatocytes, bone marrow cells, islet cells, and endothelial cells. However, the derivation of such cell types from S is not without obstacles, and hence current research is focused on overcoming these barriers. For example, studies are underway to differentiate ES into tissue-specific CMS and to eradicate their immature properties that distinguish them from adult CMS. Besides in the future becoming an important alternative to organ transplants, ES are also being used in field of toxicology and as cellular screens to uncover new chemical entities that can be developed as small molecule drugs. Studies have shown that cardiomyocytes derived from ES are validated in vitro models to test drug responses and predict toxicity profiles. ES-derived cardiomyocytes have been shown to respond to pharmacological stimuli and hence can be used to assess cardiotoxicity like torsades de pointes. ES-derived hepatocytes are also useful models that could be used in the preclinical stages of drug discovery. However, the development of hepatocytes from ES has proven to be challenging, and this hinders the ability to test drug metabolism. Therefore, current research is focusing on establishing fully functional ES-derived hepatocytes with stable phase I and II enzyme activity. Researchers have also differentiated ES into dopamine-producing cells with the hope that these neurons could be used in the treatment of Parkinson's disease. Recently, the development of ESC after somatic cell nuclear transfer of olfactory in-sheathing cells to a healthy oocyte has been recommended for neurodegenerative diseases. S have also been differentiated to natural killer cells and bone tissue. Studies involving ES are also underway to provide an alternative treatment for diabetes. For example, Damor et al. Were able to differentiate ES into insulin-producing cells, and researchers at Harvard University were able to produce large quantities of pancreatic beta cells from ES. Pluripotent. Several new studies have started to address this issue. This has been done either by genetically manipulating the cells, or more recently by deriving diseased cell lines identified by prenatal genetic diagnosis. This approach may very well prove invaluable at studying disorders such as Fragile X syndrome, cystic fibrosis, and other genetic maladies that have no reliable model system. Yuri Verlinsky, a Russian-American medical researcher who specialized in embryo and cellular genetics, developed prenatal diagnosis testing methods to determine genetic and chromosomal disorders a month and a half earlier than standard amniocentesis. The techniques are now used by many pregnant women and prospective parents, especially those couples with a history of genetic abnormalities or where the woman is over the age of 35, when the risk of genetically related disorders is higher. In addition, by allowing parents to select an embryo without genetic disorders, they have the potential of saving the lives of siblings that already had similar disorders and diseases using cells from the disease-free offspring. Scientists have discovered a new technique for deriving human embryonic stem cell. Normal ESC lines from different sources of embryonic material including morula and whole blastocysts have been established. These findings allows researchers to construct ESC lines from embryos that acquire different genetic abnormalities. Therefore, allowing for recognition of mechanisms in the molecular level that are possibly blocked that could impede the disease progression. The ESC lines originating from embryos with genetic and chromosomal abnormalities provide the data necessary to understand the pathways of genetic defects. A donor patient acquires one defective gene copy and one normal, and only one of these two copies is used for reproduction. By selecting egg cell derived from embryonic stem cells that have two normal copies, researchers can find variety of treatments for various diseases. To test this theory, Dr. McLaughlin and several of his colleagues looked at whether parthenogenetic embryonic stem cells can be used in a mouse model that has thalassemia intermedia. This disease is described as an inherited blood disorder in which there is a lack of hemoglobin leading to anemia. The mouse model used had one defective gene copy. Embryonic stem cells from an unfertilized egg of the diseased mice were gathered, and those stem cells that contained only healthy hemoglobin genes were identified. The healthy embryonic stem cell lines were then converted into cells transplanted into the carrier mice. After 5 weeks, the test results from the transplant illustrated that these carrier mice now had a normal blood cell count and hemoglobin levels. Differentiated somatic cells and ES cells use different strategies for dealing with DNA damage. For instance, human foreskin fibroblasts, one type of somatic cell, use non-homologous end joining, an error-prone DNA repair process, as the primary pathway for repairing double-strand breaks during all cell cycle stages. Because of its error-prone nature, NHEJ tends to produce mutations in a cell's clonal descendants. ES cells use a different strategy to deal with DSBs. Because ES cells give rise to all of the cell types of an organism including the cells of the germ line, mutations arising in ES cells due to faulty DNA repair are a more serious problem than in differentiated somatic cells. Consequently, robust mechanisms are needed in ES cells to repair DNA damages accurately, and if repair fails, to remove those cells with unrepaired DNA damages. Thus, mouse ES cells predominantly use high-fidelity homologous recombinational repair to repair DSBs. This type of repair depends on the interaction of the two sister chromosomes formed during S phase and present together during the G2 phase of the cell cycle. HRR can accurately repair DSBs in one sister chromosome by using intact information from the other sister chromosome. Cells in the G1 phase of the cell cycle have only one copy of each chromosome. Mouse ES cells lack a G1 checkpoint and do not undergo cell cycle arrest upon acquiring DNA damage. Rather, they undergo programmed cell death in response to DNA damage. Apoptosis can be used as a fail-safe strategy to remove cells with unrepaired DNA damages in order to avoid mutation and progression to cancer. Consistent with this strategy, mouse ES stem cells have a mutation frequency about 100-fold lower than that of isogenic mouse somatic cells. The major concern with the possible transplantation of ESC into patients as therapies is their ability to form tumors including teratoma. Safety issues prompted the FDA to place a hold on the first ESC clinical trial, however no tumors were observed. Propagation. Usefulness. The main strategy to enhance the safety of ESC for potential clinical use is to differentiate the ESC into specific cell types that have reduced or eliminated ability to cause tumors. Following differentiation, the cells are subjected to sorting by flow cytometry for further purification. ESC are predicted to be inherently safer than iPS cells because they are not genetically modified with genes such as c-Myc that are linked to cancer. Nonetheless, ESC express very high levels of the iPS-inducing genes, and these genes including Myc are essential for ESC self-renewal and pluripotency, and potential strategies to improve safety by eliminating c-Myc expression are unlikely to preserve the cell's stemness. However, N-Myc and L-Myc have been identified to induce iPS cells instead of c-Myc with similar efficiency. Utilizations. Potential clinical use. Human embryonic stem cells as models of genetic disorders. Repair of DNA damage. Adverse effect. In 1964, Louis Kleinsmith and G. Barry Pierce Jr. isolated a single type of cell from a teratocarcinoma, a tumor now known to be derived from a germ cell. These cells isolated from the teratocarcinoma replicated and grew in cell culture as a stem cell and are now known as embryonal carcinoma cells. Although similarities in morphology and differentiating potential led to the use of EC cells as the in vitro model for early mouse development, EC cells harbor genetic mutations and often abnormal karyotypes that accumulated during the development of the teratocarcinoma. These genetic aberrations further emphasized the need to be able to culture pluripotent cells directly from the inner cell mass. In 1981, embryonic stem cells were independently first derived from mouse embryos by two groups. Martin Evans and Matthew Kaufman from the Department of Genetics, University of Cambridge published first in July, revealing a new technique for culturing the mouse embryos in the uterus to allow for an increase in cell number, allowing for the derivation of ES cells from these embryos. Gail R. Martin, from the Department of Anatomy, University of California, San Francisco, published her paper in December and coined the term embryonic stem cell. She showed that embryos could be cultured in vitro and that ES cells could be derived from these embryos. In 1998, a breakthrough occurred when researchers, led by James Thompson at the University of Wisconsin-Madison, first developed a technique to isolate and grow human embryonic stem cells in cell culture. On January 23, 2009, Phase I clinical trials for transplantation of oligodendrocytes derived from human ES cells into spinal cord injured individuals received approval from the U.S. Food and Drug Administration, marking it the world's first human ES cell human trial. The study leading to this scientific advancement was conducted by Hans Kierstead and colleagues at the University of California, Irvine, and supported by Geron Corporation of Menlo Park, CA, founded by Michael D. West, Ph.D. A previous experiment had shown an improvement in locomotor recovery in spinal cord injured rats after a 7-day delayed transplantation of human ES cells that had been pushed into an oligodendrocytic lineage. The Phase I clinical study was designed to enroll about 8 to 10 paraplegics who have had their injuries no longer than 2 weeks before the trial begins, since the cells must be injected before scar tissue is able to form. The researchers emphasized that the injections were not expected to fully cure the patients and restore all mobility. Based on the results of the rodent trials, researchers speculated that restoration of myelin sheaths and an increase in mobility might occur. This first trial was primarily designed to test the safety of these procedures, and if everything went well, it was hoped that it would lead to future studies that involve people with more severe disabilities. The trial was put on hold in August 2009 due to FDA concerns regarding a small number of microscopic cysts found in several treated rat models, but the hold was lifted on July 30, 2010. History. In October 2010, researchers enrolled and administered ESTs to the first patient at Shepherd Center in Atlanta. The makers of the stem cell therapy, Geron Corporation, estimated that it would take several months for the stem cells to replicate and for the GRNOP1 therapy to be evaluated for success or failure. In November 2011, Geron announced it was halting the trial and dropping out of stem cell research for financial reasons, but would continue to monitor existing patients, and was attempting to find a partner that could continue their research. In 2013, BioTime, led by CEO Dr. Michael D. West, acquired all of Geron's stem cell assets, with the stated intention of restarting Geron's embryonic stem cell-based clinical trial for spinal cord injury research. BioTime company Asterias Biotherapeutics was granted a $14.3 million strategic partnership award by the California Institute for Regenerative Medicine to reinitiate the world's first embryonic stem cell-based human clinical trial for spinal cord injury. Supported by California public funds, CIRM is the largest funder of stem cell-related research and development in the world. The award provides funding for Asterias to reinitiate clinical development of ASTOPC1 in subjects with spinal cord injury and to expand clinical testing of escalating doses in the target population intended for future pivotal trials. ASTOPC1 is a population of cells derived from human embryonic stem cells that contains oligodendrocyte progenitor cells. OPCs and their mature derivatives called oligodendrocytes provide critical functional support for nerve cells in the spinal cord and brain. Asterias recently presented the results from Phase 1 clinical trial testing of a low dose of ASTOPC1 in patients with neurologically complete thoracic spinal cord injury. The results showed that ASTOPC1 was successfully delivered to the injured spinal cord site. Patients followed 2-3 years after ASTOPC1 administration showed no evidence of serious adverse events associated with the cells in detailed follow-up assessments including frequent neurological exams and MRIs. Immune monitoring of subjects through 1 year post-transplantation showed no evidence of antibody-based or cellular immune responses to ASTOPC1. In 4 of the 5 subjects, serial MRI scans performed throughout the 2-3 year follow-up period indicate that reduced spinal cord cavitation may have occurred and that ASTOPC1 may have had some positive effects in reducing spinal cord tissue deterioration. There was no unexpected neurological degeneration or improvement in the 5 subjects in the trial as evaluated by the International Standards for Neurological Classification of Spinal Cord Injury exam. The Strategic Partnership 3 grant from CIRM will provide funding to Asterias to support the next clinical trial of ASTOPC1 in subjects with spinal cord injury, and for Asterias product development efforts to refine and scale manufacturing methods to support later-stage trials and eventually commercialization. CIRM funding will be conditional on FDA approval for the trial, completion of a definitive agreement between Asterias and CIRM, and Asterias' continued progress toward the achievement of certain predefined project milestones. In vitro fertilization generates multiple embryos. The surplus of embryos is not clinically used or is unsuitable for implantation into the patient, and therefore may be donated by the donor with consent. Human embryonic stem cells can be derived from these donated embryos, or additionally they can also be extracted from cloned embryos using a cell from a patient and a donated egg. The inner cell mass, from the blastocyst stage of the embryo, is separated from the trophectoderm, the cells that would differentiate into extra embryonic tissue. Immunosurgery, the process in which antibodies are bound to the trough ectoderm and removed by another solution, and mechanical dissection are performed to achieve separation. The resulting inner cell mass cells are plated onto cells that will supply support. The inner cell mass cells attach and expand further to form a human embryonic cell line, which are undifferentiated. These cells are fed daily and are enzymatically or mechanically separated every 4 to 7 days. For differentiation to occur, the human embryonic stem cell line is removed from the supporting cells to form embryoid bodies, is CO-cultured with a serum containing necessary signals, or is grafted in a three-dimensional scaffold to result. Clinical trial. Embryonic stem cells are derived from the inner cell mass of the early embryo, which are harvested from the donor mother animal. Martin Evans and Matthew Kaufman reported a technique that delays embryo implantation, allowing the inner cell mass to increase. This process includes removing the donor mother's ovaries and dosing her with progesterone, changing the hormone environment, which causes the embryos to remain free in the uterus. After 4-6 days of this intrauterine culture, the embryos are harvested and grown in vitro culture until the inner cell mass forms egg cylinder-like structures, which are dissociated into single cells, and plated on fibroblasts treated with mitomycin C. Clonal cell lines are created by growing up a single cell. Evans and Kaufman showed that the cells grown out from these cultures could form teratomas and embryoid bodies, and differentiate in vitro, all of which indicating that the cells are pluripotent. Gail Martin derived and cultured her ES cells differently. She removed the embryos from the donor mother at approximately 76 hours after copulation and cultured them overnight in a medium containing serum. The following day, she removed the inner cell mass from the late blastocyst using microsurgery. The extracted inner cell mass was cultured on fibroblasts treated with mitomycin C in a medium containing serum and conditioned by ES cells. After approximately 1 week, colonies of cells grew out. These cells grew in culture and demonstrated pluripotent characteristics, as demonstrated by the ability to form teratomas, differentiate in vitro, and form embryoid bodies. Martin referred to these cells as ES cells. Techniques and conditions for derivation and culture. It is now known that the feeder cells provide leukemia inhibitory factor and serum provides bone morphogenetic proteins that are necessary to prevent ES cells from differentiating. These factors are extremely important for the efficiency of deriving ES cells. Furthermore, it has been demonstrated that different mouse strains have different efficiencies for isolating ES cells. Current uses for mouse ES cells include the generation of transgenic mice, including knockout mice. For human treatment, there is a need for patient-specific pluripotent cells. Generation of human ES cells is more difficult and faces ethical issues. So, in addition to human ES cell research, many groups are focused on the generation of induced pluripotent stem cells. On August 23, 2006, the online edition of Nature Scientific Journal published a letter by Dr. Robert Lanza stating that his team had found a way to extract embryonic stem cells without destroying the actual embryo. This technical achievement would potentially enable scientists to work with new lines of embryonic stem cells derived using public funding in the USA, where federal funding was at the time limited to research using embryonic stem cell lines derived prior to August 2001. In March 2009, the limitation was lifted. Derivation from humans. Derivation from other animals. Potential method for new cell line derivation. The iPSC technology was pioneered by Shinya Yamanaka's lab in Kyoto, Japan, who showed in 2006 that the introduction of 4 specific genes encoding transcription factors could convert adult cells into pluripotent stem cells. He was awarded the 2012 Nobel Prize along with Sir John Gordon for the discovery that mature cells can be reprogrammed to become pluripotent. In 2007 it was shown that pluripotent stem cells highly similar to embryonic stem cells can be generated by the delivery of 3 genes to differentiated cells. The delivery of these genes reprograms differentiated cells into pluripotent stem cells, allowing for the generation of pluripotent stem cells without the embryo. Because ethical concerns regarding embryonic stem cells typically are about their derivation from terminated embryos, it is believed that reprogramming to these induced pluripotent stem cells may be less controversial. Both human and mouse cells can be reprogrammed by this methodology, generating both human pluripotent stem cells and mouse pluripotent stem cells without an embryo. This may enable the generation of patient-specific ES cell lines that could potentially be used for cell replacement therapies. In addition, this will allow the generation of ES cell lines from patients with a variety of genetic diseases and will provide invaluable models to study those diseases. However, as a first indication that the induced pluripotent stem cell technology can in rapid succession lead to new cures, it was used by a research team headed by Rudolf Jaenisch of the Whitehead Institute for Biomedical Research in Cambridge, Massachusetts, to cure mice of sickle cell anemia, as reported by Science Journal's online edition on December 6, 2007. On January 16, 2008, a California-based company, StemAgen, announced that they had created the first mature cloned human embryos from single skin cells taken from adults. These embryos can be harvested for patient-matching embryonic stem cells. The online edition of Nature Medicine published a study on January 24, 2005, which stated that the human embryonic stem cells available for federally funded research are contaminated with non-human molecules from the culture medium used to grow the cells. It is a common technique to use mouse cells and other animal cells to maintain the pluripotency of actively dividing stem cells. The problem was discovered when non-human sialic acid in the growth medium was found to compromise the potential uses of the embryonic stem cells in humans, according to scientists at the University of California, San Diego. However, a study published in the online edition of Lancet Medical Journal on March 8, 2005 detailed information about a new stem cell line that was derived from human embryos under completely cell and serum-free conditions. After more than 6 months of undifferentiated proliferation, these cells demonstrated the potential to form derivatives of all 3 embryonic germ layers both in vitro and in teratomas. These properties were also successfully maintained with the established stem cell lines. Induced pluripotent stem cells. Contamination by reagents used in cell culture.